Simple diffusion does not require special membrane proteins for movement of molecules. However, other types of diffusion do require such proteins for the transport of molecules. Nonspecific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of nonspecific facilitated diffusion, these permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Some facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of a specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
The energy stored in ATP can drive membrane transport of nutrients into the cell. The largest family of energy-driven transport systems are the ABC transporters, named for their ATP binding cassettes, which are also called nucleotide binding domains. There are 2 main types of ABC transporters, influx and efflux. Influx ABC transporters carry critical nutrients such as sugars and amino acids. Efflux ABC transporters include multi-drug pumps that drive out antibiotics and toxins. Other efflux transporters secrete enzymes such as proteases and lipases out of the cell. An ABC transporter typically consists of two hydrophobic proteins that form a membrane channel and two peripheral cytoplasmic proteins with nucleotide binding domains for binding to ATP. A system that takes in a particular nutrient, or solute, uses a solute binding protein to grab the solute, either as it floats by a gram-positive microbe or as the molecule enters the periplasm of a gram-negative cell. Solute binding proteins have a high affinity for their matched solutes, so their use increases the efficiency of transport when concentrations of the solute are low. The solute binding protein directs the solute to the periplasmic face of the channel protein. The interaction with the channel protein triggers a structural change in the channel protein that is telegraphed to the ATP binding cassettes. On receiving this signal, the nucleotide binding proteins hydrolyze ATP and send another conformational change through the channel, opening up the cytoplasmic side to allow the solute to enter the cell. ADP and inorganic phosphate are released, and the transporter returns to its resting state.